Now, so far, Chapter 5 here, we've talked mostly looking at generating maybe the B-cell receptor. Now let's look at what happens when we want to expand our repertoire of immunoglobulins. We want to expand to get that 10 to the 11th potential. 100 billion, billion different antibody repertoire. Well, what we see is there's going to be 4 main processes that can help us generate this. First being combinatorial diversity. As we saw in previous lecture 5.1, it's multiple gene sequences. These multiple segments. Multiple V's, multiple J's, multiple D's, multiple C's. Another way is junctional diversity. What happens during the joining? What if during the joining of V to D or V to J, there happens to be an insertion of an A, or there happens to be a deletion of a G? Because remember, we're talking double-strand breaks here, and we're trying to join them together. There's no overlapping sequences so that we get a nice, clean fixation here. So you can end up with some slight mutations here. Now it's also going to be the different light chains going with different heavy chains. Which kappa, which lambda, going, which is it going to be? Is it going to be a kappa or a lambda joining with the H here for this B cell? And then what they refer to as somatic hypermutation within the V regions. Remember our hypervariable regions. Change an amino acid here, change an amino acid there due to 1 point mutation at the DNA level. Get a slight change here, there, within those CDR 1, 2, 3 regions. You get something completely different. The RSS sequences that we have the double strand breaks. Notice they're AGC, GC here, everything's there. RAG comes through, cuts them back, boom, boom. And now we start removing. TC, TC, and then you start adding in the palindromes. TA, TA, start adding in palindromes. Hopefully you get to a point that you can then start adding in N nucleotides, random. So between the palindrome P nucleotides and the random N nucleotides, you're going to get a part where you can get some base pairing. Here the artist drew it as just 2, but I forget what the number is. It's going to have to be a little bit more than that to get a stable base pairing, complementary strand pairing. But then after that, what you'll see is that it's stable enough that DNA enzymes can go through and fill in where it is only single-stranded. P region, N region, P region. Thing to keep in mind here at the D, the J, where we see this joining. This is going to correspond to the CDR3 region and our hypervariable region of the heavy chain. So this randomization you see in here that can be happening during the N portion, this we see contributes to the potential of a greater repertoire. Is it always going to work? No. If it decreases the affinity of the antibody, decreases the affinity of the B cell receptor to an antigen, that B cell will never be selected for. It will slowly die out as its shelf life goes through. If it causes for a non-functioning antibody, thus a non-functioning receptor, because everything downstream from here is now out of frame. B cell receptor won't form. this B cell will die off. Only correctly formed B cell receptors, the correctly forming heavy chains, those B cells are the only ones that will be allowed to mature. Those are the only ones that will get the signal to become plasma cells and thus antibody producing. We see that the T cell receptor, because remember this is also based, immunoglobulin domain based. We talked about this chapter 4. We see some of the same things. Numerous V's, numerous J's, numerous C's. The thing here, remember it's an alpha and a beta chain for these guys, but look at the numbers. The potential different V's, 70 to 80. The different J's, 61. Heavy chain, or the beta chain, which we would think of as heavy chain, almost. 52 B's, numerous D's, numerous J's, numerous C's. Rearrangement is going to be roughly the same. See, this is also part of the reason why we refer to these as immunoglobulin-like domains, because somewhere there was an ancestral domain that rearranged similar to these, and then it spread, and it was kept, it was maintained evolutionarily, and now we see B cell, T cell receptors, as well as our immunoglobulins using some of the same processes. I mean, look at this. Germline for the alpha, V1 to ends. Some V joins with some J, joins with the C. You get the alpha chain. Look at the beta. There's a reason why I mistakenly said, hey, look, it's the heavy chain. Because look at it. V's, D's, J's, and C's. This is quite similar to what we see with the heavy chain. It's just here there's only one constant domain. There's not two more. It is a cytoplasmic tail transmembrane region we see here. Our variable regions, again, are going to be V and J for alpha, are going to cross over V, D, and J for the beta. Look again, some of the same things. RSSs, our same signal sequences. We see them here flanking the V's, the D's, the J's for the alpha, beta T cells. There's also special types of T cells known as the gamma deltas. They have a slightly different role in the immune response that we're not going to talk about here. But again, look, V's, D's, J's, 23 and 12, 23 and 12, that will interact for recombination. When you look at what's going on here, if you learn it with the B cells, it holds true relatively with the T cells when it comes to the recombination and generating such a vast repertoire. How you can go from a few T cells and then have T cells, a T cell for every epitope you will ever be exposed to. Same with the B cells. B cell, a few types of B cells, and by the time you have a healthy, mature immune system, you have a B cell generating an antibody for almost every antigen you could ever be exposed to. And look at the numbers. The variable, 40. Kappa lambda, 70. Beta, 50. Alpha, 70. So it looks like somewhere evolutionarily, Kappa lambda would have been similar to the alphas because the numbers add up. Except here when you look at the joining. 6, 5, or 4 if you're Kappa or lambda. 61 if you're alpha. Number of variable gene pair, V gene pairs. How many different ways you can do it? 1.9 times 10 to the 6th, 5.8 times 10 to the 6th. The J, the junctional diversity, you start factoring in those, about 3 times 10 to the 7th, 30 million. 2 times 10 to the 11th, so that's 200 billion. So you can, just in the VDJ segments, generate a repertoire of 50 trillion different VDJ combinations. Not all of them are going to work. Not all of them will be functional. Not all of them will ever actually have an antigen that will trigger a response by that B cell. Over here, 10 to the 18th. I don't even know how many billions, trillions that is. But still, not all of them are going to work. Not all of them are going to function. But the repertoire, the potential is there. And what's interesting is when you look at CDR3 alpha, CDR2 beta, 3 beta, 2 alpha, 1 alpha, when you look at what's going on here, it's these variable regions. The areas where you're going to have the joining is most often going to be the area that you're going to see interact with the epitope in the T cell receptor. So, by having the P palindromic sequences and the N randomness, it increases our potential for something here in these strands that will be able to interact with all potential epitopes you can be exposed to. We also see that the receptor potential can be generated just by sheer rearrangement of genes. Specifically, we're going to see this with the gamma, the delta, but you see this sometimes in the alpha. What we see happen is that the genes will be cut out, moved around. Not much going on. We saw this with the B cell receptor. You're going to have looping around. You're going to rearrange the sequence that they're in, not the actual DNA AGCT sequence, but V1, V2, followed by 3, 4, and will now become V1, V2, and then straight to J. That type of rearrangement. Because as we see here, we can loop around, cut out a bunch of these intervening segments, these intervening loci. If they're not there, then the potential for them to ever be expressed is now gone. So you would go directly from V alpha, whatever this one is, to J alpha, whatever that one is. The intervening V's, D's, J's, and C's no longer possible. This also feeds into our